Hi guys, this is Luke and welcome to my channel. So today, what we are going to talk about is the nature, scope, and types of development. We will also discuss the process of gametogenesis and its hormonal regulation. We will also uncover the process of fertilization and early developmental stages in relation to established germ cell and organ rudiments. And lastly, we will gonna talk about organogenesis of the nervous system and the integumentary system. Every life on Earth originated in a single cell. Multicellular organisms such as animals and plants undergo series of complex processes of development starting from a single cell, forming the actual adult organism all throughout life. These developmental processes that an individual organism undergoes during its lifespan is what we know as ontogenetic development. There are 6 major phases of ontogenetic development, namely gametogenesis, fertilization, cleavage formation, gastrulation, organogenesis, and growth and development respectively. Gametogenesis is the formation of haploid sex cells, meaning that these cells contain half the number of genome from the parent cell. In most animals, including humans, it has 2 types: spermatogenesis, which is the formation of sperm in males, and oogenesis, which is the formation of mature ovum in females. The second phase of ontogenetic development is fertilization. This includes the fusion of male and female sex cells, forming a diploid zygote. This means that a zygote contains 2 pairs of genome coming from both parent cells. Next is the cleavage formation, which is basically an early process of embryological development. This is the differentiation of zygote into few cells, giving rise to embryo. There is no growth in cell size at this phase. Instead, it divides without changing its original size, which is the size of an egg cell. Gastrulation comes after the cleavage formation. This is the process of the formation of 3 primary germ layers, which is the ectoderm, endoderm, and the mesoderm germ layers. Ectoderm gives rise to our skin and nervous system. Mesoderm form the muscles, circulatory system, bones, kidneys, reproductive system, and other organs found in our body cavity. While endoderm forms the lining of our internal organs such as the walls of our digestive tract, lungs, liver, and etc. Organogenesis, from the word itself, is the process of these 3 germ layers developed into different organs of our body. It basically forms the organ rudiments or the undeveloped parts that make up a specific organ. The last phase of ontogenetic development is growth and differentiation. Organ rudiments produced in organogenesis undergoes further maturation of its physiochemical properties observed in an adult individual. Now let's talk about the cell cycle. The cell cycle is one of the significant physiological processes that occurs in an organism. Cell cycle is divided into 5 phases. It begins with the G1 phase, which is the primary growth phase where young cells grow physically larger. Next is the synthesis phase where cells synthesizes its DNA inside the nucleus. And then the G2 phase, also known as the secondary growth phase, where all the organelles necessary for cell division is created and duplicated. These 3 phases belong to the interphase, wherein it serves as a preparation for cell division. There are 2 types of cell division. We have mitosis, or the division of somatic cells, and the other one is meiosis, which occurs to produce sex cells. The last phase of the cell cycle is known as cytokinesis, where cleavage furrows separates the parent cell into 2 daughter cells. Now let's go back to the 4th phase of the cell cycle. In mitosis, also known as the karyokinesis, is divided into 5 subphases with an acronym of PPMAT or PPMAT. This stands for prophase, where nuclear envelope and nucleolus disintegrates and spindle fiber starts to appear from the centrosome. The second P stands for prometaphase, where spindle fiber attach to kinetochores found at the center of the condensed chromosomes. M for metaphase comes next, where chromosomes arrange themselves in the middle region of the cell known as the equatorial plate. A for anaphase, where sister chromatids are pulled away from each other toward opposite poles, which in return elongates the whole cell. And lastly, T stands for telophase, where the divided chromosomes are surrounded in each nuclear envelope that forms. Then the next one is the cytokinesis, or the division to form daughter cells. Now take a note of this. Not all somatic cells undergo this PPMAT phase. There are cells that are arrested in the G0 phase and do not undergo interphase unless initiated, and some stays permanently in this phase. The next type of cell division is known as meiosis, which is totally different compared to mitosis. It has two rounds of cell division, the PMAT1 and the PMAT2. Instead of producing two diploid identical cells, it produces 4 haploid unidentical sex cells. I have mentioned a while ago about the two types of gametogenesis. Spermatogenesis occurs in males, specifically in the tightly coiled seminiferous tubules, once an individual reaches his puberty. It begins with a mitotic division to form spermatogonia, or the undifferentiated germ cells formed in the seminiferous tubules wall. Meiotic phase of spermatogenesis has two subphases, PMAT1 and PMAT2, that forms 4 haploid spermatozoa. And the last phase, called spermeiogenesis, which modifies the shape of spermatozoa into a functional one by removing most of its cytoplasm, giving rise to a streamlined sperm. It takes about 60 to 75 days of sperm production. After a streamlined sperm was formed, it is transported into epididymis for further maturation and ready for ejaculation. The whole process of spermatogenesis is initiated through the help of luteinizing hormone, which interacts with the Leydig cells to produce testosterone. It stimulates spermatogonium to differentiate into primary spermatocyte. And follicle-stimulating hormone interacts with Sertoli cells to provide support and nutrition for sperm proliferation and development. Oogenesis, on the other hand, begins even before an individual is born. The whole process is not a continuous, but rather it is intermittent, or have intervals at certain point of a female's lifespan. It occurs in the ovary. After birth, primary oocyte is already formed in the female baby, but it is arrested in prophase 1 of meiosis 1. Only after the first menstruation that the meiosis is completed. Each month, a single ovum is released in the fallopian tube, but this time, it is arrested at metaphase 2 of meiosis 2. The complete division of a female sex cell will occur once it is fertilized by a sperm, and if not, it goes along with the disintegrated endometrium each month. Overall, the process produces 3 polar bodies, or a non-functional ova, and 1 functional ovum. Follicle-stimulating hormone regulates the formation of follicles in the ovary, which houses the early underdeveloped oocytes. Luteinizing hormone, on the other hand, signals the primary oocyte to complete meiosis 1 where it left off. The corpus luteum is a remnant of ovarian follicle, releases progesterone that thickens the uterine lining for the implantation of the ovum. We've also given a brief overview about the fertilization. It is a process by which sperm and egg fuse together, forming a zygote, which occurs at the fallopian tube. Fertilization has 4 major stages. Number 1 is the preparation, which involves capacitation and acrosome reaction. Second is the binding. Next is the fusion, and followed by activation. Capacitation is the adjustment that a semen does as it enters the vagina with regards to the vaginal pH, making the sperm be able to find its way to the egg. Acrosome reaction is the release of enzymes at the acrosome of sperm to forage and successfully enter the egg's membrane. Sperm binding occurs when sperm pass through the zona pellucida. Both egg and sperm membrane bind with each other through the aid of receptor proteins present in their membranes. After the binding, sperm and egg pronucleus starts to appear and fuse together to combine the chromosomes they contained. This process completes the meiosis II of an egg where it was arrested. Then the last stage is activation, which initiates cortical reaction that basically hardens the egg's outer layer, preventing polyspermy to occur. Earlier, I discussed about the gastrulation and organogenesis. These processes occurs right after the fertilization. This time, we are going to talk about how our nervous system is formed. Ectoderm germ layer is the origin of our nervous system. It has two major divisions, the central nervous system and the peripheral nervous system. The organogenesis of our nervous system begins at the process called neurulation, which forms the neural tube that occurs at the 4th week of embryological development. Neural tube gives rise to the brain and spinal cord. Neural crest was also formed during neurulation, which gives rise to peripheral nervous system. Notochord, although plays an essential role in the development, however, is not a part of the final nervous system. A process called vesiculation is also part of the development of the nervous system. During this process, the 3 primary vesicles were formed. These are the prosencephalon, mesencephalon, and rhombencephalon. Eventually, prosencephalon gives rise to secondary vesicles called the telencephalon, which forms the cerebrum and the globus pallidus. And the other one is the diencephalon that forms the thalami. Mesencephalon, on the other hand, remains as is, which is a rudiment of midbrain. And the third one is the rhombencephalon, which gives rise to metencephalon that becomes the pons and the cerebellum. And the last one is the myelencephalon, which gives rise to medulla. Our skin is made up of 3 major layers. The epidermis, or the outermost layer, that serves as a barrier to the external environment. The next one is the dermis, where blood and lymph vessels are found, as well as the different glands such as the sebaceous gland. And lastly, the hypodermis, which connects the underlying muscles to the skin. Along with the nervous system, our skin was also derived from the ectoderm, which begins at the 4th week of embryological development. All skin layers originates from somite, specifically the sclerotome. Ectoderm, also known as the basal layer, undergoes differentiation, giving rise to the periderm. Keratinization is a process in which the cytoplasm of periderm is replaced by keratin. These keratinized cells ascend into the surface and sheds off, which eventually replaced by new cells. The basal layer eventually becomes the stratum germinativum. Replacement of the peridermal cells continue to occur until week 21. At birth, all epidermal layers are present. Starting from the top, these are the stratum corneum or the horny layer, stratum granulosum or the granular layer, stratum spinosum or the spinous layer, and lastly, the stratum germinativum or the germinative layer. Under lies the ectoderm are the mesenchyme cells. These cells gives rise to the dermis. And that's the end of this video. For more educational videos like this, please click on subscribe and hit the notification bell below. Enjoy the rest of your day and don't forget to put a smile on your face.